rubidium, and that gas is actually placed to the whole environment, the room in which the person is, has to be nullified in terms of magnetic fields. So there are coils on the left and the right hand side, and the subject is in between, and these coils are tuned so that at the electrodes you have no magnetic field. So here you can, instead of having the bunker, you do it without the bunker, but you still need to do something about the magnetic field of the Earth. Here you have to send currents through those coils, so there is no magnetic field in the location you want to record. And these rubidium electrodes are being aligned. There is a laser beam going through it and a photodetector that picks up a measure related to the opacity of the vial here. And you see it in the plot here, the bottom plot. The intensity is the highest when there is no magnetic field. But as you can guess, if there is a magnetic field coming from brain activity, then these rubidium atoms will be deflected in their path, and this will make the vial more opaque. And this is picked up by the sensor, and then you see in the plot that the intensity is going down. So it's sort of optical principle that is used, the relation, the sensitivity of the optics being related to the magnetic field. So that's interesting, of course. And of course, the development need to proceed to make it even more compact and more useful. And here you see a plot here of the frequencies. It's like a frequency scaleogram as a function of time. The red line indicates the onset of a stimulus, and then you see suddenly in the frequency range between 50 and 70. Yeah, of course, the rubidium gas is special. In between 50 and 70 hertz, you see this yellow orange-like activity past the onset of the stimulus, and you see some blue stuff in the lower frequency range. This is the mu band, and the yellow stuff is the gamma band. So here you see some combination, and that's of course due to motor activity. The subject was performing motor activity, the mu band activity goes down, and the gamma band activity goes up. And the gamma band is of course very interesting because that's related to activity, the neural activity of large groups of neurons. And the gamma band activity is a bit of a holy grail because with EEG it's not that evident to record it, and most labs don't even attempt to record it, just cut it away, they say it's rubbish. So, but with OPM you can record seemingly some gamma band activity, which makes the technique for us, for BCI purposes, very, very interesting. So we are here collaborating with this lab, and we will be hopefully publishing the first paper on brain-computer interfacing with OPM. We have been also looking at some simulated data to see what is the spatial resolution of this technique. And you see on the bottom, the S indicates SOPM, S is simulated. And you see the dipole here is much more localized compared to the squid case. Squids remember the sensors that are used in the traditional MAG setup. You see a much better spatial resolution. For that, the reason for that is of course, that these electrodes are very, very close to the scalp, and much to the case of MEG. And this is a pretty nice advantage, and that can be exploited in many ways. And this is what we also try to do with our BCI. Just a moment, I need to do something. 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 Okay, so this was slide 31. We move to slide 32. We come then to technology that you probably have seen in a clinical environment. You have all sorts of so-called scanners. You have CT scanners, you have PET scanners, MR scanners, and what more. So what is an MR scanner? Well, it's also looking at, it's using magnetic stuff. And you can recognize by the very bulky donut-like shaped scanner. So it's very thick, and the opening is pretty small. If you look carefully, the person is horizontally, and there is a sort of brace here. Indeed, the head needs to be fixated when you record brain activity, because it's extremely sensitive to all movements. So your head should not be moving, because otherwise you get a lot of corruption, the signal. Okay, so this is the environment. So slide 33, it is shown how it is being used. So this kind of scanners are used to take a picture of organs, of tissue, of bones, bone tissue, and so on. For example, in case of the brain, if there was damage to the brain because there was a hemorrhage, a stroke, for example, then the neurologists want to figure out what is the extent of the damage. A neurosurgeon wants to use this kind of imagery to prepare for surgery to remove a brain tumor, for example. So this is a static image relating to the anatomy of the organ, in this case the anatomy of the brain. This is traditional MRI, what is being used for. In case of so-called functional MRI, it's looking at brain function. Here you want to pick up a dynamic picture of the brain, dynamic related to the activity of changes in activity of the brain. So the magnetic principle is being used here in the case of the anatomy and static imagery, because tissue has different properties also with respect to in terms of magnetic properties, and these differences can be picked up. Like fatty tissue is typically having another to have a lot of water molecules in there, and this gives another magnetic property than for other type of tissue. And when you do functional MRI recordings, you are looking a little bit at a different principle. We'll explain it in a moment. Looking at slide 34, you see the difference between that static image, which is of very high quality, and functional image, which is a bit blurry. Okay, so this is what it looks like. The reason is also very simple. With a static image, you can collect, nothing is moving here, so to speak. You can collect data for a prolonged time period. You can summate all these images that you have collected, and you get rid of noise and stuff. But in case of that dynamic picture, every snapshot is a snapshot of ongoing activity, and the ability to handle noise is much more restricted. So moving to the next slide, you see the principle of fMRI is a sort of natural contrast agent, as they call it. It's called the BOLD signal, blood oxygenation level dependent signal, and it's an indirect measure of neural activity. Indeed, when neural activity of a particular region in the brain goes up, then there will be fresh oxygen-rich blood flowing. The volume of that will increase. There will be more flowing through that area than before, and the properties of that oxygen labeled blood is different from low oxygen blood, and this difference can be picked up in terms of differences in magnetic properties, and that is the fMRI signal. So this is what is being picked up. So it's not neural activity, but something that is related to neural activity, namely it's related to blood. So if you go through the slide 34, there are some pictures popping up. So you have, as I said, a time series of brain images when you do fMRI. On slide 35, you see a bit the setup. In case of psychophysics experiment, the person is in the coil.